action. Alright, this is Mr. Babatunde Folorunshaw, your instructor for practical MIC 211, that is Student Microbiology. Now, the reason why I'm making this video is to ensure uniformity. Uniformity because I'm not the only one that's going to mark your manual, so let us be on the same page. Now, so I'm starting from page 63 and page 64, that is 63 and 64. So that's page 63 and 64, worksheet 12. The question is on staining fungal hyphae. So what I want to highlight there is very in question 1b, the question says that states the group the two organisms provided belong. So we did not observe that in our manual, but basically the two groups that we usually observe are Rhizopus and Rhizopus or mucor, which belong to Zygomycotina or Zygomycetes. Then also Penicillium, that belongs to what? Ascomycetes and Ascomycotina. So the two groups so you just say, okay, either Rhizopus or mucor, the first one, then that belongs to the group of Zygomycetes. Then Penicillium, the second group, that is Ascomycetes. So then another important question is that in question number 5, you were asked to give the types of reproduction found in fungi, giving an example in each case. So you have to give us an example. So because we have 4 groups, we have Ascomycetes, Enteromycetes, Basidiomycetes, Zygomycetes. So at least you'll be able to give us 4, then you give us examples of each. So that is just that page. So if once you answer that page correctly, you are good to go. So you don't expect, I don't want to see any report in that particular page. Alright, so we now go to page 60. Page 60 is talking about capsule stain. Yes, so capsule stain. So for page 60 is talking about capsule stain. So draw and label your observation. So your observation for the capsule stain, you draw and label it. Your course rep for your department is going to send that to you, so you draw and label what you have seen. Please, a lot of you are asking the question, do you paint to differentiate do you color to differentiate gram positive bacteria from gram negative bacteria? How about that question should not be coming from you. This is biology, okay? No shading, no painting. Alright, so that is for that one. So you answer question 1A, 1B, 2, 3A, 3B, then to 5, 4 to 5, yeah. So you answer that one, that is for capsule stain. So that's another place that you're answering. Alright, so we did not do a lot of this, so I'm flipping those pages. Please don't do what we did not do in class. So the next thing that we did was spore staining or endospore staining. That's what we did. Alright, so for spore staining or endospore staining, you see that in page 51 to page 51 to page 54. Alright, so you record your observation carefully in drawing and note. So record your observation carefully in drawing. That means you draw and make notes. What do you see in that image that I've sent to you? Alright, draw what you have seen. Alright, then there are other questions from 2 to 4 and okay, I'm just trying to see whether there's anything I need to explain here. Alright, so you answered that. So that means for page 52 to page 54, you provide your answer to that particular place. So we move on to the next question. The next question is called differential staining. That's exercise 10, and that is from page 49 to 50. So now, so this one, they said to make drawing and notes with peculiar reference to colors, shape, size, etc. of bacteria. So I've sent you like 4 different gram positive, gram negative rod, and so you note they are different. You make a drawing of each one. You now say that what you should note their color. So you tell us the color, tell us the shape, tell us their size. So you tell us the color, not show us the color. So that means you are not allowed to color anything. Alright, so there are a lot of questions under it that you need to answer. Please, you have to give us, don't just answer, make sure you make research. and don't just copy and paste from the internet. All right, so you will know whether you are in the lab or not. So that is for what. So you are answering from from page 49 to 50 for differential staining. That is exercise 10. All right, so the next one is, so please, as I'm opening it, take your manual and open and tick them what you're supposed to do. So the next one is talking about exercise 9, that is staining of microorganism. So the question starts from page 6 to page 44 to page 44 to page 45 first. Then also there's a follow-up question. Please. And that follow-up question, okay, I think that's all. So that is, you only have for that, you have worksheet 9 from page 44 to page 49. Now draw and label the different kind of bacteria observed in the exercise above. What exercise are we talking about? Okay, so that is this gram staining. So the same bacteria that you have drawn, because of limited space, you can draw gram negative and gram positive, gram negative rod and gram negative cocci in this particular place, and gram positive and gram, so in the previous place. So just distribute them. Now, so then you now answer the adjoining question. Okay, so what conclusion can you draw from this experiment? So when they asked you what conclusion can you draw from the experiment, the conclusion that you can draw from the experiment is that gram positive bacteria stains, you state the color, gram negative bacteria stains, so you state the color of what you have done in the staining procedure, of that is staining of microorganism. Then we now go to worksheet 8, that is pour plate method of isolation, spread plate method. Okay, sorry. So worksheet, yeah, well, yes, worksheet 8. Yes, we are still in worksheet 8. So worksheet 8 is all about isolation of pure culture. That's what worksheet 8 is all about. So it talks about streaking, yes, streak plate method of isolation, pour plate method, and spread plate method, and extinction diffusion method. So basically, so you're going to answer worksheet 8 question, which is in page 38 to page 41. Okay, so you answer them correctly. All right, you answer them correctly. Take your time to make research on what you have done. Some of them we have mentioned in the class, but some of them they are in the lab practical class. Some of them were mentioned in your theoretical class. Now it is very important for you to find out what is, understand it, and put it in your own words, because some of these questions are going to come out in your words in your exam. Practical questions are going to come out in your exam, so take your time to understand them. So that's what, for that one, you're going to answer that question. Then also worksheet 7, exercise 7, that is morphology of algae. Most of you, some of you have, some, the other group, the last group, have not done this, but the last group that came in the last lab did the morphology of algae. Now in the morphology of algae, it is in page 31 to 32. That's where you are going to answer. Okay, page 31 to page 33, 31 to 33. Now in page 31 there's a space in page 31, so you're going to make your drawing in that particular place. That is, draw the cell, observe, and label fully. So you're going to draw the cell, observe, and label fully. Then you answer question 1 to question 5, which is on page 32 to page 33. So you answer those questions in that particular place, in that particular manner. So that is on exercise 7, morphology of algae. Then yeast, exercise 6, we did not do that, so don't touch it at all. But if you have touched it, no problem, we are not going to mark it. So we did not do anything on yeast. Please note that I'm not going to mark it, and my colleagues are not going to mark that one too. Then we talk about morphology and cultural characteristic of fungi. All right, if you can remember that lab, we used PDA and NA, and you came to do your counting and observe the morphological characteristic. So that is worksheet 5. So that means you are going to answer from page 25 to page 20, page 25 to page 26. So the first question says, draw and label the observed fungi under the microscope. Actually, we did not observe this fungi under the microscope, the one we did the last time, but there was a practical that we did, staining, IFAS staining, that you observe under the microscope. Okay, that was the last practical that we did. We observe under the microscope, so you draw that one here, then Then you answer the question number 2. Okay, from number 2 to number 9. So you draw them, you answer them in that particular manner. Alright, then you now talk about worksheet 4, morphological and cultural characteristics of bacteria. So that is number 1, question number 1 to question number 5. Alright, so in this particular place, you are not asked to draw. Alright, so that is what we are going to answer in the particular place. Okay, so that is what we are observing. Then we now talk about morphology and cultural characteristics of bacteria, but I don't think we are don't bother. Alright, don't bother to draw anything. But in question number 2, that is page 22, worksheet 4, page 22, this question says that what appearance of colony of the same bacterium differ depending on the variety of factors. Mention 3. No, sorry, that's not what I want to say. Okay, question number 2. Examine the given plates and perform a count and colonial characterization of the microorganism. So count. He said you should perform a count here. So you count. If this place number 2 is not enough, if question number 2, page 22, if the space is not enough to do the count, move to page 21 and do the count in that space. Then tell us the morphological or colonial characteristic. Is it elevated? What's the pigmentation and all that? So you can just give us like 4 of them in question number 2. Alright, so that is that. Then also the question that is worksheet 3, media preparation. So media preparation is worksheet 3. So you're going to answer question on page 17 to page 19. So page 19. Okay, so you're going to answer that question in that particular manner. Then you now go to exercise 2. So what's exercise 2? Exercise 2 is about media again. Okay, so you answer those questions. They're just theoretical questions and the composition, about the composition of media. You are going to answer page 12 to page 13. You are marking that. Very, very important that you are marking that. Then you now go to the first page, that is basic apparatus and uses. That is what we use. So you are going to describe and state the use of the following. So you are describing them and are stating the uses of the following apparatus. So note, you describe and state the uses. Now after the question starts from page 3 to page 9, so you have it. So that's where you have the question. So you are going to draw inside the drawing book all this material that you have said and attach it. But your drawing, please note, your drawing does not, you don't need to tell us the function again in the drawing. Just draw and give it a name and label as appropriate. So when I say label as appropriate, that means you cannot, there's nothing to label in a McCartney bottle. But when you're talking about autoclave, you should be able to label. When you're talking about anaerobic jar, you should be able to label. When you're talking about microscope, you should be able to label. So not all of them are going to have, you're not going to label all of them, but some needs to be labeled. Okay, so you are not going to be penalized for that strictly. You are not going to be penalized if you're not labeling. But your drawing, you are going to look at your drawing. Okay, is it a biological drawing? Is it did you draw with a smooth, a thin line continuous pen or a thick one? If you draw with a thick pencil, you are going to lose mark. If you draw with a thin pencil, you're going to get mark. If you don't shade, you're going to get mark. If you shade, you're going to lose mark. If you don't, your arrow for labeling, your line for labeling does not touch what you are pointing to directly, horizontally, you're going to lose mark. So all those things are observed while you are drawing. So this is the format that I want you to use to answer the questions. So this is just to act as a guide so that everybody will be on the same page. Then for those that are just joining, by next week, by the grace of God, because you have missed some of the practical that we have done, so I'll just try and do a video on the practical that you have done, so that you can be able to have an idea of what we are doing. So I want you to just follow this format, and everything that I've mentioned are only those that I'm going to mark. So take note of that.